And we're back. We've made our messenger RNA. We're going to take that information and turn it into a protein. So we have to introduce a new character here, and that's transfer RNA, or tRNA for short. Transfer RNAs hold on to amino acids, and they have a little area at the bottom called an anticodon, and that actually reads the codon of the messenger RNA. So here it is. So here's the area that holds on to the messenger RNA. You can see it is one big long strand. Down here is an anticodon, which reads the codon, and then so areas of hydrogen bonding are here, 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 and here. This is a three-dimensional structure. It's probably actually what it looks like. Here is kind of a schematic that has the business ends where the amino acid is. Whoa. And where the codon is. I'm sorry, the anticodon. One of the unsung heroes is amino acyl tRNA synthetase. What that does is it takes the amino acids and it takes some energy, reads a transfer RNA, the enzyme does that, and then links the amino acid with the transfer RNA. So here's our amino acid. We link it to the enzyme, we phosphorylate it, the transfer RNA fits into the kind of the active site. So this bottom, if it doesn't read very well, the transfer RNA won't fit in there, and then boop, on its way. It's said to be charged. It's an amino acyl tRNA, so it's a tRNA with amino acid on it. So with ribosomes, we have the small and the large subunits. They also have RNA in them, called ribosomal RNA, or rRNA for short. So far we got tRNA for transfer RNA, messenger RNA, and then rRNA. Here. So if you look at it, we got our large subunit, small subunit. We got the A, P, and E site. A is for where the amino acids are at, P is where the polypeptide or protein's at, and E is for the exit site. And you'll see what kind of goes on in a minute. Here's a summary. A holds the amino acid, P holds the polypeptide, E is where it goes away, exit. 3 stages to this: initiation, elongation, and termination. So initiation is the start, elongation is where kind of everything's happening, and the termination is the end. All 3 of these stages need what are called factors, or so you have initiation factors, elongation factors, and termination factors. They all kind of push this process along. And we're not really going to focus on them so much except for termination. But if they're not present, this process is not occurring. So initiation, we have the small subunit. It ends up linking up with this transfer RNA, and then it binds with the messenger RNA. All right. Then the large subunit lands on top of everything. Boom. That is initiation. It's not a whole lot to it, but it gets everything started up. Then we get to elongation, where everything is kind of occurring. So here we've got like a transfer RNA in the P site, and this is kind of mid-process, really, if you think about it. It's holding onto a polypeptide. Then this transfer RNA that's holding onto an amino acid lands into the A site. All right, so there's like a cavity in the middle of this ribosome, and they can kind of insert themselves through the top and then exit kind of towards the back. So it lands in the A site, and then there's like basically a peptide bond is made between these two, between the two like amino acids here. So a peptide bond is made, and now for a real brief moment, the transfer RNA that's sitting in the A site is holding onto the polypeptide, and then the ribosome moves over one codon, and then this now is holding on the polypeptide. This transfer RNA here now is in the E site, so it exits. Whoops. it exits. Interestingly enough, that actually takes ATP or GTP, takes energy to get it to exit. They actually had to pry off the transfer RNA from the hydrogen bonds between the tRNA and mRNA. And then we can start this process all over again, because then we have the tRNA, it's sitting in the P site, it's holding onto a polypeptide. This this transfer RNA that has an amino acid lands in the A site. They make this peptide bond. The ribosome moves over. Whoops. We've got an exiting of the transfer RNA, and it just keeps on going until we get to stop codon. So stop codon signals the system to stop. All right. So there's actually tRNA for the stop codon. It lands in the site. It grabs onto the polypeptide chain, and then the termination factor is inserted into the A site, or it's called a release factor, because after the release factor goes in there, everything gets released. The polypeptide is released from the tRNA, and then the small and large subunits of the ribosome are going to break off, and then the release factor breaks off too, and that's termination, the end of this whole process. But at this point, now you've got your polypeptide chain or your protein, and you're good to go. What's interesting in bacteria, transcription and translation actually occur simultaneously. So as transcription in the bacteria is occurring, the ribosomes jump onto the mRNA, and you've got a bunch of them, as you can see from this electron micrograph or this picture here. They all just kind of jump on, and they're making their proteins, and then they go on with their life, or their happy little way. Here you can see just a big mess of what's happening in the eukaryotic cell. So here we've got our DNA, we've got our RNA polymerase. It's making our mRNA. It gets edited, then it leaves the nucleus, goes wherever. We've got our, so our translation would start over here. We're making our polypeptide chain, it falls off, and then we have our protein. The cell lives happily ever after.